Can viruses change their antigenicity? And if so, how can they change? Firstly, we need to clarify some associated terms. Genotype refers to the genetic constitution of an organism. Phenotype refers to the appearance of an organism, which are produced by genotype in cooperation with the environment. Mutation is an inheritable change in the genotype. Y-type virus denotes the original virus from which mutants are derived and with which the mutants are compared. Mechanisms of variation of viruses include mutation, gene recombination and reassortment, and interaction of viral gene productions. 1. Mutation. The conditional lethal mutant is because of mutation. Mutants are lethal under one set of conditions, non-permissive conditions, but that yield normal infectious progeny under other conditions, permissive conditions. For example, temperature-sensitive mutant. This mutant can grow at low temperatures, but not at high temperatures. Oral poliomyelitis vaccine is a temperature-sensitive mutant. So when the children take the vaccine, make sure it should not be taken with hot water, because the hot temperature may inactivate the vaccine. Another one is a host range mutant. This mutant differs from the wild type in the targets that can be infected. 2. Gene recombination and gene reassortment. The classic recombination mechanism is that the nucleic acid strands break, and a part of the genome of one parent is joined to part of the genome of the second parent. Gene reassortment. If a virus has a segmented genome, and if two variants of that virus infect a single cell, progeny variants can result with some segments from one parent, some from the other. Look at this diagram. The pink virus and the green virus infect the host cell at the same time. So the progeny viruses could carry pink gene and the green gene. That is gene reassortment. Typical example of gene reassortment is flu virus. When human flu virus and non-human flu virus, for example, avian flu virus, infect one respiratory epithelial cell, the reassortant virus may contain some segments from human virus and some segments from avian virus. And the reassortant virus may have the ability to cause severe disease and can be transmitted from person to person. That could be a big disaster. 3. Interaction of viral gene productions. Complementation refers to the interaction of viral gene products in cells infected with two viruses, one or both of which may be defective. It results in the replication of one or both under conditions in which replication would not normally occur. The example is HDV and HBV. With the help of HBV, the HDV can invade and replicate in liver cells. Phenotypic mixing. If two different viruses infect a cell, progeny viruses may contain coat components derived from both parents and so they will have code properties of both parents. Phenotypic mixing, no change in genome, but sensitivity to antibody neutralization is altered. If the genome is encased in a completely different protein code, this extreme example of phenotypic mixing may be called transcapsidation. This progeny virus is also known as pseudotype. Two publications associated with pseudotypes. People used pseudotypes as a tool to make vaccines. The first paper showed the pseudotyped influenza A virus as a vaccine for the induction of heterotypic immunity. Similarly, for the second paper, pseudotype virus-like particles to generate antibodies. Viruses can change their antigenicity by mutation, gene recombination and reassortment, and complementation and phenotypic mixing. How to classify the viruses? Family of viruses. Family is based on morphology, genome structure, and strategies of replication. Suffix viridae, and should be in italic. Genus. Genus is based on physical, chemical, or serological differences. Suffix virus. Example: Orthomictoviridae, influenza virus. Orthomictoviridae is a family name. Influenza virus is a genus name. Based on biological properties, the viruses can be classified as DNA viruses, RNA viruses, and retroviruses. There are enveloped DNA viruses and naked capsid DNA viruses. Enveloped DNA viruses include pox virus, herpes virus, and herpesdina virus. And the naked capsid DNA viruses are polyoma, papilloma, adeno, and parvoviruses. Parvovirus is a single-stranded DNA virus. The RNA viruses include positive RNA, negative RNA, double-stranded RNA, and the retro RNA viruses. Most of them are enveloped. Just picornavirus and calicivirus are naked RNA viruses. And the reovirus has double capsid, but no envelope. The viruses can be classified by the mode of transmission. They are viruses transmitted through respiratory route, viruses transmitted through fecal-oral route, and others. Others include hepatitis virus, herpes virus, hemorrhagic fever virus, arbovirus, and HIV. In this course, we will learn about the viruses based on these classifications. Here are the viruses we will cover in this course, medical virology. Subviruses. Viroids are essentially circular RNA molecules that have been associated with several plant diseases. They do not encode proteins or possess a capsid. Prions are infectious protein particles. This pathogen does not contain any genome, just the misfolded proteins. But it can induce a very serious disease, mad cow disease. We will have one section on it. So far, we have learned the structure, replications, genetics, and classifications of virus. The keywords are virus, basic structure, self-replication, temperature-sensitive mutant, abortive infection, defective virus, complementation, interference, gene recombination and reassortment, pseudotype. Now, a quiz for you. A 13-month-old boy presents with a 4-day history of fever and watery non-bloody diarrhea in winter. On physical examination, he appears dehydrated. Which of the following describes the genome of the most likely organism? A. Segmented negative sense single-stranded RNA. B. Non-segmented positive sense single-stranded RNA. C. Segmented double-stranded RNA. D. Double-stranded DNA. E. Single-stranded DNA. To answer this quiz, firstly, you need make a preliminary diagnosis, and then to recall the knowledge about the genome type. You may not answer correctly now, but do not worry about that. When we learn about each virus, we will get to know all of them.